हेलो फ्रेंड्स, राना अकेडमी में आपका स्वागत है। हमने स्टार्ट किया है 12th क्लास की एनसीईआरटी की वन शॉट सीरीज़ केमिस्ट्री की। उसमें हमने 13 यूनिट तक कंप्लीट किया है। स्टार्ट करते हैं 14 यूनिट, जो कि हमारा बायोमॉलिक्यूल्स। द लिविंग सिस्टम ग्रोज, सस्टेन्स एंड रिप्रोड्यूसेस itself. The most amazing thing about a living system is that it is composed of non-living atoms and molecules. The pursuit of knowledge of what goes on chemically within a living system falls in the domain of biochemistry. Taking living systems are made up of various complex biomolecules like carbohydrates, proteins, nucleic acids, lipids, etc. Protein or food. Proteins and carbohydrates are essential constituents of our food. These biomolecules interact with each other and constitute the molecular logic of life processes. In addition, some simple molecules like vitamins and mineral salts also play an important role in the functions of organisms. Structures and functions of some of these biomolecules are discussed in this unit. Carbohydrates. Carbohydrates are primarily produced by plants and form a very large group of naturally occurring organic compounds. Some common examples of carbohydrates are cane sugar, glucose, starch, etc. Most of them have a general formula C6 H2O 3 times and were considered as hydrates of carbon, from where the name carbohydrate was derived. For example, the molecular formula of glucose C6H12O6 fits into this general formula. C6H2O 6 times, but all the compounds which fit into this formula may not be classified as carbohydrates. For example, acetic acid CH3COOH fits into raminoles. C6H12O2 O5 is a carbohydrate but does not fit in this definition. A large number of their reactions have shown that they contain specific functional groups. Chemically, the carbohydrates may be defined as optically reactive polyhydroxyaldehydes or ketones, or the compounds which produce such units on Hunter lysis. Some of the carbohydrates which are sweet in taste are also called as sugars. The most common sugar used in our homes is named as sucrose, whereas the sugar present in milk is known as lactose. Carbohydrates are also called as saccharides. Greek saccharum, saccharon means sugar. Classification of carbohydrates. Carbohydrates are classified on the basis of their behavior on hydrolysis. They have been broadly divided into following three groups: monosaccharides, oligosaccharides, and polysaccharides. First, a monosaccharide, a carbohydrate that cannot be hydrolyzed further to give a simpler unit of polyhydroxy, aldehyde, or ketone is called a monosaccharide. About 20 monosaccharides are known to occur in nature. Some common examples are glucose, fructose, ribose, etc. Oligosaccharides, carbohydrates that yield 2 or to 10 monosaccharides units on hydrolysis are called oligosaccharides. The oligosaccharides, etc. Depending upon the number of monosaccharides they provide on hydrolysis, amongst these the most common are disaccharides. The two monosaccharides units obtained on hydrolysis of a disaccharide may be on same or different. For example, one molecule of sucrose on hydrolysis gives one molecule of glucose and one molecule of fructose, whereas gives one molecule of only glucose. Polysaccharides, carbohydrates which yield a large number of monosaccharide units on hydrolysis are called polysaccharides. Some common examples are starch, cellulose, glycogen, gums, etc. Polysaccharides are not sweet in taste, hence they are also called non-sugars. The carbohydrates may also be classified as either reducing or non-reducing sugars. All these carbohydrates which reduce swellings, solution and tolerance reagent are referred to as reducing sugars. All monosaccharides further, whether aldose or ketose, are reducing sugars. Monosaccharides are further classified on the basis of the number of carbon atoms and the functional group present in them. If a monosaccharide keto group, if a monosaccharide contain an aldehyde group, it is known as an alkylose, and if it contains a keto group, it is known as ketose. Number of carbon atom constituting a monosaccharide is also introduced in the name as in is evident from the examples given in table 14.1. 14.1 table, different types of monosaccharides. Glucose, preparation of glucose. Glucose occurs freely in nature as well as in the combined form. It is present in sweet fruits and honey. Ripe grapes also contain glucose in large amounts. It is prepared as follows: from sucrose, cane sugar. If sucrose is boiled with dilute HCl and H2SO4 in alcoholic solution, glucose and fructose are obtained in equal amounts. From starch, commercially glucose is obtained by hydrolysis of starch by boiling it with dilute H2SO4 at 393 Kelvin under pressure. Structure of glucose. Glucose is an alcohol and is also known as dextrose. It is the monomer of many of the larger carbohydrates, namely starch, cellulose. It is probably the most abundant organic compounds on Earth. It was assigned the structure given below on the basis of the following evidences. Its molecular formula was found to be C6H12O6 on prolonged heating with HI. It forms CN-hexane, suggesting that all the 6 carbon atoms are linked in the straight chain. The glucose reacts with the hydroxyamine to form an oxime and adds a molecule of hydrogen cyanide to give cyanohydrin. These reactions confirm the presence of carbonyl group C double bond O in glucose. Glucose gets oxidized to 6 carbon carboxylic acid, gluconic acid, and reaction with a mild oxidizing agent like bromine water. This indicates that the carbonyl group is present as an aldehyde group. Acetylation of glucose with acetic anhydride gives glucose pentacetate, which contains the presence of 5 OH groups. Since it exists as a stable compound, 5 OH groups should be attached to different carbon atoms. On oxidation with nitric acid, glucose as well as gluconic acid both yields dicarboxylic acid, saccharic acid. This indicates the presence of primary alcoholic group in glucose. The exact spatial arrangement of different OH groups was given by Fischer after studying many other properties. Its configuration is correctly represented as 1. So gluconic acid is represented as 2, and saccharic acid has 2. 3. Glucose is correctly named as D positive glucose. D before the name of glucose represents the configuration, whereas plus represents dextro rotatory nature of the molecule. It should be remembered that D and L have no relation with the optical activity of the compound. They are also not related to letter delta and T. D and T, see in its 10. The meaning of D and L the notations is as follows. The letters D or L before the name of any compound indicates the relative configuration of a particular stereoisomers of a compound with respect to configuration of some other compound, a configuration of which is known. In the case of carbohydrates, this refers to their relation with the particular isomers of glyceraldehyde. Glyceraldehyde contains an asymmetric carbon atom and exists in two and anatomic enantiomeric isomers forms as shown below. Plus isomer of glyceraldehyde has of configuration. It means that whereas its structural formula is written on paper following specific conventions which you will study in higher classes. The OS group lies on the right hand side in the structure. All those compounds which can be chemically correlated to D positive isomer of glyceraldehyde are said to have D configuration, whereas those which can be correlated to L. Now, some isomer of glyceraldehyde are said to have L configuration, and in L isomer OH groups is of on left hand side as as you can see in the structure. For assigning the configuration of monosaccharides, it is the lowest asymmetric the carbon atom as shown below, which is compared as in plus glucose. OH on the lowest asymmetrical carbon is on the right side, which is comparable to glyceraldehyde. So plus glucose is assigned D configuration. Other symmetric carbon atom of glucose are not considered for this comparison. Also, the structures of glucose and glyceraldehyde is written in way that most oxidized carbon, in this case CHO, is at the top. Cyclic structure of glucose. The structure of glucose explained most of its properties, but the following reactions and facts could not be explained by this structure. Despite having the aldehyde group, glucose does not give shifts test, and it does not form the hydrogenous sulfide. Addition product with NaHSO3. The pentacetate of glucose does not react with hydroxylamine, indicating the absence of a CHO group. Now, glucose is found to exist in two different crystalline forms, which are named as alpha and beta. The alpha form of glucose 419 Kelvin is obtained by crystallization from concentrated solutions of glucose at 303 Kelvin, while the beta form 423 Kelvin is obtained by crystallization from hot to saturated aqueous solution at 371 Kelvin. This behavior could not be explained by the open chain structure. For glucose, it was proposed that one of the OH group may have found that glucose forms a six-membered ring in which OH at C5 is involved in the ring formation. This explains the absence of CHO A group and also exists of glucose in two forms as shown below. These two these two cyclic forms exist in equilibrium is with open chain structures. The two cyclic hemiacetal forms of glucose differ only in the configuration of the hydroxyl group at C1 called anomeric carbon. The aldehyde carbon before cyclization, such isomers, that is alpha form and beta form, are called anisomers. The six-membered cyclic structure of glucose is called a pyranose structure, alpha and beta. In analogy with pyron, pyron is a cyclic organic compound with one oxygen atom and five carbon atoms in the ring. The cyclic structure of glucose is more correctly represented by Howard's structure as shown in below. A structure is an important ketoxidose hexose. It is obtained strong along with glucose by the hydrolysis of disaccharide sucrose. It is a natural monosaccharide found in fruits, honey, and vegetables in its pure forms. It is not used as a sweetener. It is also an important ketohexose structure of fructose. Fructose also has the molecular formula C6H12O6, and on the basis of its reactions, it was found to contain a ketonic functional group as at carbon number 2 and 6 carbons in straight chain, as in the case of glucose. It belongs to D series and is a laboratory compound. It is appropriately written as D-glucose. Its open chain structure is as shown. It also exists in two cyclic forms, which are obtained D-fructose by the addition of OH at C5 to the C double bond O group. The ring has thus formed is a five-membered ring and is named as furanose. With analogy to the compound furan. Furan is a five-membered cyclic compound with one oxygen and four carbon atoms. Disaccharides. You have already read that disaccharides on hydrolysis with the dilute acids or enzymes yield two molecules of either the same or different. The monosaccharides, the two monosaccharides are joined together by an oxide the linkage formed by the loss of water molecules. Such a linkage between two monosaccharide units through oxygen atom is called a glycosidic linkage. The disaccharides if the reducing groups of monosaccharides, that is aldehydic and ketonic groups, are bonded. These are non-reducing sugars. That example sucrose. On the other hand, sugars in which these functional groups are free are called reducing sugars. For example, maltose and lactose. Such sucrose, one of the common disaccharides of is sucrose, which on the hydrolysis gives equimolar mixture of D glucose and D fructose. Now these two monosaccharides are held together by glycoside linkage between C1 and D alpha D glucose and C2 beta D glucose. Now since the reducing groups of glucose and fructose are involved in glycosidic bond formation, sucrose is a non-reducing sugar. Sucrose is dextrorotatory, but after hydrolysis it gives dextrorotatory glucose and laboratory fructose. Since the laboration of fructose 92-92.4 degree is more than the dextrorotation of glucose 52.5 degree. Now the mixture of laboratory and thus hydrolysis of to leave, and the product is named as invert sugar maltose. Another disaccharide maltose is composed of two alpha D glucose units in the which chlorine of the one glucose is linked to C4 of another glucose unit 3 unit 2. The free aldehyde group can be produced at C1 of second glucose in solution, and it shows reducing properties, so it is reducing sugar. Lactose, it is more commonly known as milk sugar. Since this disaccharide is found in milk, it is composed of beta D galactose and beta D glucose. The linkage is between Cl and of galactose and C1 and C4 of glucose. A free aldehyde group may be produced at C1 of glucose units, hence it is also reducing sugar. Lactose. Polysaccharides contain a large number of monosaccharide units joined together by glycosidic linkages. These are the most commonly encountered carbohydrates in nature. They mainly act as a food storage on structural materials. Starch is the main storage for polysaccharide of plants. It is it is the most important dietary source of human beings. High content of starch is found in cereals, roots, tubers, and some vegetables. It is a polymer of alpha glucose and consists of two components, which constitutes about 50 to 20% of starch. Chemically, amylose is a long unbranched chain with 200 to 1000 alpha D plus glucose units held together by C1 to C4 glycosidic linkage. Amylopectin is insoluble in water and consists of about 80 to 85% of starch. It is a branched chain polymers of alpha D glucose units in which chain is formed by C1 to C4 glycosidic linkage, whereas branching occurs by C1 to C6 glycosidic linkage. Cellulose occurs exclusively in plants, and it is the most abundant organic substance in plants kingdom. It is the predominant constituent of cell wall of plant cells. Cellulose is a straight chain polysaccharide compound only of beta D glucose units which are joined by glycosidic linkage between C1 and of now one glucose unit and C4 of the next glucose unit. Glucose, the carbohydrates are stored in animal body is as glycogen. It is also known as animal starch because its structure is similar to myelopectin and is rather more highly attached. It is present in liver, muscles, and brain. When the body needs glucose, enzymes breaks the glycogen down. The glycogen is also found in yeast and fungi. Importance of carbohydrates. Carbohydrates are essential for life in both plants and animals. They form a major proportion of our food. Honey has been used for a long time as an instant source of energy by Vedas and Ayurvedic system of medicine. Carbohydrates are used as storage molecules as starch or in plants and glycogen in animals. Cell wall is of bacteria and plants is made up of cellulose. We build furniture, etc. from cellulose in the form of wood and clothe ourselves with cellulose in the form of cotton fiber. They provide raw materials for many important industries like textiles, paper, lacquers, and brewers. The aldopentoses was D-ribose and D-ribose. 6 section 14.5.1 class 12 are present in nucleic acid. Carbohydrates are found in biosystem in combination with many proteins and lipids. Proteins, amino acids, classification of amino acids, structure of proteins, globular proteins, fibrous protein, globular protein, denaturation of proteins, enzymes, mechanism of enzyme action, vitamins, classification of vitamins, nucleic acids, chemical composition of nucleic acids, structure of nucleic acids, biological functions of nucleic acids, and then summary. Carbohydrates are optically active polyhydroxy aldehydes or ketones or molecules which provide such units on hydrolysis. They are broadly classified into three groups: monosaccharide, disaccharide, and polysaccharides. Glucose, the most important source of energy for mammals, is obtained by the digestion of starch. Monosaccharides are held together by glycosidic linkages to form disaccharides or polysaccharides. Proteins are the polymers of about evenly 20 different alpha amino acids which are linked by peptide bonds. 10 amino acids are called essential amino acids because they cannot be synthesized by our body. Hence must be provided through diet. Proteins contains only alpha vitamins. Alpha amino acids are called simple proteins. The secondary or tertiary able to performs their functions. This is called denaturation of proteins. Enzymes are biocatalysts which speed up the reactions within biosystems. They are very specific and selective in their section, and chemically, all enzymes are proteins, and vitamins are accessory food factors required to the diet. They are classified as fat soluble A, D, E, and K, are water soluble B group and C. Deficiency of vitamins leads to many diseases. Nucleic acids are the polymers of nucleotides, which in turn consist of base, the pentose sugar, and phosphate moiety. Nucleic acids are responsible for the transfer of characters from parents to offsprings. There are two types of nucleic acids, DNA and RNA. DNA contains of 5 carbon sugar molecules called 2-deoxyribose, whereas RNA contains ribose. Both DNA and RNA contains adenine, guanine, and DNA is a double strand, whereas RNA is a single strand molecule. DNA is the chemical basis of heredity and have the coded message of proteins to be synthesized. In the cell, there are three types of RNA and RNA, mRNA. mRNA, rRNA, tRNA, which actually carry out the protein synthesis in the cell. सो ये 12th का हमारा चैप्टर है केमिस्ट्री का, जिसको हमने वन शॉट सीरीज में कवर अप किया है. पहले से देखते हैं. बायोमोलिक्यूल्स 14th यूनिट है हमारा. कार्बोहाइड्रेट्स, क्लासिफिकेशन ऑफ कार्बोहाइड्रेट्स, मोनोसैकेराइड्स, प्रिपरेशन ऑफ ग्लूकोज, स्ट्रक्चर ऑफ ग्लूकोज, साइक्लिक स्ट्रक्चर ऑफ ग्लूकोज, fructose, structure of fructose, disaccharides, polysaccharides, importance of carbohydrates, proteins, amino acids, classification of amino acids, structure of proteins. fibrous protein, globular protein, denaturation of proteins, enzymes, mechanism of enzyme action, vitamins, classification of vitamins, vitamin A, vitamin B1, vitamin B2, vitamin B6, vitamin B12, vitamin C, vitamin D, vitamin E, vitamin K, nucleic acids, chemical composition of nucleic acids, structure of nucleic acids, biological functions of nucleic acids, एंड देन समरी बायोमॉलिक्यूल्स के. सो यहाँ पर हमने बहुत ज़्यादा शॉर्ट अप में कवर अप किया इस चैप्टर्स को, जो कि आपके रिवीजन में बहुत ज़्यादा बेनिफिशियल होगा. प्लीज़ इस वीडियो को ज़्यादा से ज़्यादा शेयर कीजिएगा. प्लीज़ लाइक शेयर एंड सब्सक्राइब. थैंक यू सो मच फॉर वॉचिंग.